Okay, so hi everyone, my name is Selket Delafontaine and welcome to this ESID Junior Journal Club. So first of all, before I forget, to remind you to certainly apply to go to the ESID summer school. I went last year and it was an amazing experience, which I can certainly recommend to everyone. You should apply before the 31st of January. Well, I'll subscribe and try to apply for it. And then I am very excited to introduce our speaker for today. She's, Marta Dafne Cabañero, and she's coming from sunny Valencia. She's an internal medicine resident at the University and Polytechnic Hospital in Valencia, and she's also doing her PhD in clinical immunology. And so today we have the unique opportunity to be introduced to Marta's research, and she's going to present why there's an important connection between dysimmunity in CVID and altered microbiota. And she's also going to accentuate why specific bacterial biomarkers can predict more severe CVID phenotypes. So without further ado, Marta, good luck and thank you very much for sharing your valuable insights today. The floor is yours.
So thank you, Selket, for this introduction and this presentation, and also thank you to ESID for giving me this opportunity. I am very happy to be here with you, showing you one part of my PhD project, which is about microbiota in CVID. And hopefully I will be presenting it in 1 month, so I hope you like it. And also I would like to thank you to all the team that has been taking part of this study, the researchers, the physicians, and everyone. So this part of my PhD is about dysimmunity in CVID and how this dysimmunity is associated with alterations in oral, respiratory, and intestinal microbiota. So to start with, I would like to introduce a little bit about what is CVID. It's a primary immunodeficiency heterogeneous group, as you all know. It's a group, it's not actually a disease itself, because nowadays we are discovering that CVID is a group of diseases that are under the same umbrella. They have a similar clinical phenotype, but we don't really know the cause of this disease. Its prevalence is very low, 1 out of 25,000 patients to 1 out of 50,000 patients. And they are mostly distributed in developed countries, as you can see here. This is not because CVID chooses to be in developed countries, it's because in undeveloped countries there are no registries, and we should pursue that in undeveloped countries they also register CVID. The etiology of this disease is hypogamma. It's usually a hypogammaglobulinemia. It courses with decreased IgG together with decreased IgA as IgM sometimes, after excluding other causes. But as you can see in the right part of the screen, the diagnosis of CVID is not only an analytical diagnosis, it also includes several clinical features, like, for example, autoimmune manifestations, granulomatous disease. I'm going to put this down. Granulomatous diseases, unexplained polyclonal proliferations, and other type of autoimmunity or autoinflammation. So that's why under this heterogenic pattern, the group of Chapel in 2008 divided CVID into distinct clinical phenotypes. Those presenting polyclonal lymphocytic infiltration, those with autoimmunity, those with enteropathy, those with malignancy, and those showing only infections. These patients that show only infections is because they have this hypogammaglobulinemia, but the other patients, they have these manifestations because their immune system also has a dysregulation. So it's a dysimmunity, what they have. And usually in the literature, these are called like severe CVID, which is something important for this study. So basically, as I have just said, we don't really know the cause of CVID patients, but during these last 22 years, researchers have been discovered several genes, several monogenic mutations that can explain CVID patients in 1/3 of the cases. We still don't know the cause of the other 70% of the cases. That's where microbiota may play a role. These genetic discoveries has been so important that in this past 2022, the International Union of Immunological Societies have established CVID not as a disease itself, but as a phenotype, as you can see here, Common Variable Immunodeficiency phenotype, in which, according to the mutation, they have been writing down several different syndromes. But still, 70% of the patients, we don't really know what's the cause. And many researchers think that there's multiple genetic and environmental risk factors may interact together. And some studies recently have shown that there's a link between an altered microbiota and the development of autoimmune diseases like, for instance, lupus, Sjögren, and other autoimmune diseases. So to start, what is microbiota? First of all, it's symbiotic. They are symbiotic communities of microorganisms such as bacteria, viruses, fungi, archaea, and protozoa that are colonizing specific ecological niches, mainly the skin, the oral cavity, the lungs, gastrointestinal tract, and also the bladder, which has been discovered recently. Symbiotic means that there's a relationship between the host and between these microorganisms. So we provide bacteria with some benefits, and this bacteria provides us with other benefits. These bacteria have co-evolved. They have turned into specialized bacterial communities that are in charge, basically, of helping us with digestion, with vitamin synthesis, also, very importantly, with pathogen defense and immune development, and also even in some last recent research, they also related microbiota with brain development. So it's very, very important. And as you may know, this microbiota is very dynamic and very influenceable. It's not the same, our microbiota now in this very moment, than the microbiota we will have in 10 years. And that depends on diet, on lifestyle, on age, sex, host genetics, and very importantly, and a key treatment of our CVID patients in antibiotics and immunosuppressants, which will be discussed also lately. Also, I would like to introduce the concept of dysbiosis, which is an imbalance in microbiota composition. And that's in some research, they try to relate this dysbiosis with the appearance of autoimmunity diseases and also with CVID. And what we also tried to look for was for pathobionts, which are specific species that can predict or are very related with several diseases, like for example in CVID. How do we do it? First of all, so basically we do a PCR and a sequencing of the region B3, B4 of the minor subunit of the 16th, basically ribosomic RNA of bacteria, which is a region that is highly conserved in in bacteria, and is enoughly small to be sequenced also easily. So that's why it's very interesting. And what we basically measure is alpha diversity indexes or beta diversity indexes, but in our study it's alpha diversity. This alpha diversity, what measures is richness and evenness of the population. So richness is the number of species that form part of our microbiota, and evenness is not only the number of species, but the number of bacteria that are in each species. So for instance, if I have in my microbiota Streptococcus pneumoniae, Staphylococcus aureus, blah, 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 richness will be the number of species, and evenness will be the number of Streptococcus pneumoniae, the number of Staphylococcus aureus, and like that. It's a little bit hard to explain, but it's these two indexes. In general, a good microbiota or a healthy microbiota will be a microbiota with a high richness index and a high evenness index. And this richness and evenness, to make it a little bit even more complicated, is measured by Tau1 index and Shannon index, which are the two indexes that are measured in this study. So Tau1 is a measure of richness and Shannon of evenness. And there are also other indexes like beta diversity, gamma diversity that are compare the microbiota between several populations. So basically the concepts are Tau1 and Shannon are the two most important things of every study in microbiota. So this interest on microbiota and CVID started in 2016 when the group of Jorgensen basically discovered that there were a higher level of serum LPS, lipopolysaccharide, in blood of CVID patients. The lipopolysaccharide is a part of the wall of bacteria. So they thought that maybe this lipopolysaccharide came from bacteria that were being translocated from a leakage in the gastrointestinal tract of these patients. And that's when every person in the PID community started to think about microbiota. In fact, in this study, they discovered that yes, there was a reduced alpha diversity in CVID patients. And since then, as you can see in the table, many studies were looking at this same fact. So everyone basically established that yes, there was a lower alpha diversity here, in the next one also. But what is very interesting is that they didn't analyze the species level, at the species level. So they basically stopped at Streptococcus, not Streptococcus mitis, not Streptococcus pneumonia. So they didn't check for the species level of this bacteria. That is something we try to make. And very interestingly, every study on this was made in the gastrointestinal tract, not in other niches, except for one study that was made in the oral cavity that related also a decreased alpha diversity indexes in CVID, but again, not analyzing at species level. So we questioned ourselves, what happens with the other niches? What happens with pulmonary or respiratory microbiota, with the oral cavity, etc. We checked that the scientific community was having a lot of interest on respiratory microbiota, on interstitial lung diseases, for instance, also on sarcoidosis, which are a bit, they resemble a bit of CVID, especially on the granulomatous lymphocytic interstitial lung disease that happens in some CVID patients. And also interestingly, there is a gut-liver-lung axis, which means that if I have a respiratory infection, these bacteria produce some inflammatory markers that can alter microbiota also in gastrointestinal tract. And they were the same way, but the other way around. And also there's an influence of pulmonary microbiota in the oral cavity. So everything is connected. That it's something that internists we really like, and also pediatricians and everything. So we basically ask ourselves, would it be possible to relate some specific microorganisms with the clinical phenotypes of CVID? Could microbiota be used to predict CVID patients that will develop this immune complication? Or is there a role of the microbiological niches in the immune regulation of CVID? So we perform a cross-sectional study on the primary immunodeficiencies unit of my department of internal medicine at my hospital. And we try to include all the patients that we were following, more than 18 years old, that accomplished ESID registry working definition. And we also included patients that were under prophylactic antibiotic therapy and that were being treated also with immunosuppressants, which both of them alter microbiota. And we excluded patients that were taking also antibiotics, but not for a prophylactic use, but for a treatment use. We perform a retrospective revision of all these variables that you can see here in the right part, the spleen disease, liver disease, lung disease. Why? Because we wanted to classify them in these two groups that I mentioned, really regarding this Chapel classification, the classic classification of CVID. So we included 17 patients in the infectious disease group that, as you know, they have a good prognosis because you can give them immunoglobulin replacement therapy and also antibiotics, and they have a good prognosis. And we included 24 patients in the other subgroup, the dysimmune CVID subgroup, that show polyclonal lymphocytic infiltration, intestinal enteropathy, autoimmunity, or malignancy, and had a worse prognosis. We extracted saliva, sputum, and feces of these patients were extracted, collected these samples. And we also compared and have samples of control people, healthy people, of saliva, sputum, and feces. We perform a DNA extraction, and we did sequencing of this 16S subunit, and then we perform a biostatistical analysis. So first of all, going through the results, as you can see here in the patients, the healthy controls did not show any comorbidity, and the infectious only CVID group, they didn't show any other comorbidity, but one patient developed lymphadenopathies due to an infection. It was not lymphadenopathies related to polyclonal infiltration. And another patient had a vasocellular carcinoma in the skin and was removed, so it was not a malignant neoplasia. And the rest of the dysimmune CVID group, you can see here all the comorbidities, lymphadenopathies, splenomegaly, and etc. that confer a bad prognosis. So going through the indexes that I have told you before in order to start this study. First of all, you can see here in the right part of the screen here, when we compare the indexes of the saliva of these patients. So there were no significant differences in the global biodiversity in saliva. This, in the upper part, you can see the Shannon index, and in the lower part, you can see the Tau1 index. So Shannon, evenness, and Tau1, richness. In sputum samples, going to the other part of the screen, the species evenness was significantly lower in dysimmune CVID patients than compared to healthy controls. Here is the dysimmune index, the dysimmune CVID patients, their alpha index, their Shannon index, and here the controls. You can see that it's decreased. That probably it's talking about this dysbiosis on the respiratory microbiota of the dysimmune CVID patients. And regarding their fecal samples, the richness of the dysimmune CVID patients was lower compared to infectious only CVID patients, and also compared to controls. So that again talks about the dysbiosis in patients with CVID and dysimmunity. But as I said, we didn't want just to study these indexes and stay just in the global diversity. We wanted to search for pathobionts that could predict which patients with CVID could develop dysimmunity and which patients not. So we performed this predictive model depending on the number of bacteria. So in the first part, the respiratory samples, as you can see, a total of 5 to 7 species here. If you go up, you can see that they predict the CVID phenotype with more than 95% of accuracy. That means that if I am in the clinics, I am in front of a patient with CVID of our study, and I perform a test on their respiratory samples, and I find this bacteria, these 5 to 7 bacteria, I can predict that this patient has dysimmunity with a 95% of accuracy. That's what it means in our study. Still, we will have to do some external validation tests in order to know if this has a potential diagnostic role. In fecal sample, 7 species had an accuracy of 86%. And in oral sample, 5 species showed an accuracy of 92%, which is quite high. So, which are these bacteria? what are the protagonists of this model. They are listed here in this table. In the oral microbiota, this list, in the pulmonary microbiota, this list, and in gastrointestinal microbiota, also this list. So what did we do in order to know this? First of all, we performed a multivariate analysis, which is called Boruta algorithm, to know the predictive capacity of each bacteria in the model. So for instance, Bayonella tobetsuensis, which is here in pink, has a higher predictive capacity than Streptococcus cristatus, all together in the same sample. We didn't find a pathobiont, one unique pathobiont. It's the union of all these bacteria. And regarding the univariate analysis we perform, which is called ANCOMBC, we perform this univariate analysis in order to know the species frequency of CVID patients compared to ICVID patients, to the ones that show the only infections. So the bacteria that are in green were significantly higher in CVID patients. The ones that are in red are less frequent in CVID patients, and the ones that are in gray didn't show any difference between both groups. And now, regarding what we concluded from all of this, which is a lot of data. First of all, this is the first study that is analyzing the oropharyngeal, respiratory, and fecal microbiota, and at the species level, like going to the last part of the classification. And we also try to analyze that regarding the phenotypes, regarding the severity of the CVID. Global diversity is lower in the sputum and in the stool samples of patients with CVID and dysimmunity. And this is talking about a dysbiosis as a biomarker of immune dysregulation, and talks us about a less resilient microbiome. It's less able to adapt to changes, and yes, basically talks us about this dysbiosis. We developed a model that basically includes 5 to 7 species and is able to predict CVID patients' phenotype with more or less 90% of accuracy. And this has a potential role in diagnosis. As I told you, first of all, we will have to do a lot of external validation in other CVID populations to check if this is really like that. But in our study, at least with our population, is like this. And we didn't find a unique pathobiont. We find, as you can see, a lot. And looking up to other studies in other diseases, in lupus, in Sjögren, and in other things, the most important things that have a clinical sense are this list of bacteria: Bayonella, Prevotella, Streptococcus, and Alistipes. But as you can see, it's not species. They are not species, they are genera. So that's why it's very important to know what the species it's exactly. So Bayonella, Prevotella, Streptococcus, and Alistipes are found in CVID patients, are also found in CVID patients with a severe phenotype, and are also found in autoimmune disease. So they talk us about inflammation and dysbiosis and dysregulation. And for instance, regarding Streptococcus, the species level is very important because, for instance, Streptococcus mitis is related with inflammation, but other type of Streptococcus are present in healthy microbiota. So that's why it's very important to keep on doing research on this. And this study has limitations, a lot, as I can tell. First of all, CVID patients that were included are very, very heterogeneous. This limits a lot the possibility to generalize this to other CVID patients and to other primary immunodeficiencies research. Also, these changes we see in their microbiota, are they a cause or a consequence of the disease? We really don't know. What we should do is animal models. We should change microbiota in animal models with the pattern that we have seen in this article, and wait to see what happens, to see if they develop CVID, to see if they develop CVID with dysimmunity, to know if basically it's a cause or a consequence of this disease. Also, this is a single center study. Microbiome and virome has not been analyzed, and we should do it in the future. These species are very low abundance, and they are very fragile. There can be a lot of changes with time, and also important limitation is the use of antibiotics and of immunosuppressants. These patients are under antibiotic prophylactic therapy, and they are taking immunosuppressants. We cannot remove these treatments because we will take a lot of risk in the clinics and in the revolution. But it's a reality, like it's their microbiota in this very moment, in that moment where we collected the samples. And also sputum samples are contaminated with saliva because we didn't perform a bronchoalveolar lavage to know the bacteria that are exactly inside their lungs. They are a sputum sample. So in conclusion, to conclude, this systemic bacterial dysbiosis, as reflected in altered microbiota profiles in fecal, oral, and respiratory samples, could be implicated in the immunological dysregulation that is associated with severe cases of CVID. And in case you want to know more, you can check the article that has been published in Clinical Immunology in October. And I will be happy to answer any questions you may find. And thank you so much for your attention.